When we talk about diploid organisms, we always say one set of our chromosomes come from our mom and one set of our chromosomes come from dad. And that begs the question, where is all this variation come from? Why is it that with billions of people in the world, unless two individuals are identical twins, they are never going to be exactly carbon copies of one another? Well, we have that courtesy of sexual reproduction and meiosis. So we're going to talk about the three different ways in which meiosis and sexual reproduction leads to so much variation in sexually reproducing organisms. So here it is. This is a family. There are, let's see, 1, 2, 3, 4, 5, 6, 7 children, but none of them are either... is it 7? Did I count right? 1, 2, 3, 4, 5, 6, 7, 8 children. None of them are either carbon copy of their dad or carbon copy of each other. So how does that happen? There are 3 different mechanisms. One is crossing over. We already talked about crossing over in some way when we talked about meiosis. Crossing over talks about the event during prophase 1 when homologous chromosomes pair up and exchange genetic material with one another. So here it is, a pair of homologous chromosomes sitting next to one another. During prophase 1 of meiosis, they pair up and exchange genetic material through this structure, which is called chiasmata. And then when we separate them during anaphase 1, we get 2 new chromosomes. These new chromosomes don't look anything like what we started with. Now, for example, with respect to human beings, there are 23 pairs of chromosomes. Even if one crossover event happens per chromosomes, you can imagine how many different combinations of chromosomes we're going to have. And the reality is that there's not only one crossover event that happens. Actually, there are multiple crossover events that happen throughout the homologous chromosome. So that leads to a lot of variations and mix and matches of the genetic material that a sperm or egg gets, which is used. So crossing over, we're going to start here with 2 pairs of homologous chromosomes. Before meiosis happens, they duplicate. Each of them receive a sister chromatid, and then the chromosomes line up pairwise, and exchange of genetic material happens between these homologous chromosomes. Then you get different combinations after meiosis 1, and during meiosis 2 you separate the sister chromatids, and then you get 4 different types of cells from one original cell. So by the time sperm or egg are produced, the chromosome composition in terms of the different pieces that come from different parents, it kind of doesn't look like what you had before. So one sperm can have genetic material from both the mother and the father. The other mechanism for producing variation is independent assortment, and that has to do with the way homologous chromosomes line up at the metaphase plate. So imagine you have 2 pairs of homologous chromosomes, and one set, let's say, that comes from the father is blue, and the other set that comes from the mother is red. Now, when the homologous chromosomes pair up and line up together during the metaphase, you can either get all of the father's chromosomes sitting on one side of the metaphase plate and all of the mother's chromosomes that came from the mother line up on the other side. So then during metaphase 1, when you separate the homologous chromosomes, by the time meiosis 1 is over, you get only blue chromosomes here and only red chromosomes in there. And then As a result of the second division, you're going to get gametes that have only blue chromosomes and gametes that have only red chromosomes. Notice that we are ignoring crossing over. We are assuming that crossing over isn't happening, just to make this mechanism entirely clear. Okay? So that's one way in which these homologous chromosomes can sit on the metaphase plate. But where does the word independent assortment comes from? It comes from the fact that the way this pair, this top pair, let me bring my laser pointer. The way this top pair sits in the metaphase plate is totally independent of how the other pair sits. So what happens is that, okay, if you look at the top pair in the second situation, they're sitting still in the same arrangement, blue on the left, red on the right. But then these two can flip. They can flip their position, and then now you can have a red on the left for the little chromosome and blue on the right. And then when anaphase 1 happens, you can see now you have one blue chromosome and one red chromosome, unlike here. And then the same thing here. So then you're going to get entirely different gametes as a result of this arrangement. So because the way homologous chromosomes line themselves up on the metaphase plate independent of one another, you can get all sorts of combinations. Now, consider this. In our example, we are looking at two pairs of chromosomes, right? So we have two pairs of chromosomes, and each of them can sit in its own way. So then 2 to the power of 2. Okay, that's 4. So we get 1, 2, 3, 4 different combinations. So we get about, in this case, even if we don't take into account crossing over, we get 4 different combinations. But consider human beings. We have homologous pairs, but how many homologous pairs do we have? We have 23. 2 to the power of 23 is about, I don't have space here, it's 8 million, close to 8 million. 8 million. I'm not going to write million, just understand that this is million, okay? So just from one meiotic in one person, let's say one woman or a man, there is a possibility to produce 8 million different combinations of gametes. And you can imagine how astronomical that number is. And that's why no matter how many children a family has in their lifetime, they're not going to have 8 million children. So for one family, there is no way that 2 children are going to be identical to one another unless they're identical twins. Finally, the last mechanism has to do with the way fertilization happens, the way sexual reproduction happens. The sexual reproduction depends on sperm that finds an egg, that fertilizes it, and gives you a new individual. Now, there are multiple, as we know, multiple millions of sperm cells that are produced that enter into the body of the woman, and then there is an egg that's going to be fertilized. But out of these million, randomly one of them is able to enter the egg and fertilize the egg. But this process of choosing which sperm fertilizes the egg is totally random. And what type of egg is released each month from a woman's body is also random. So when you have so many different combinations and you match them up randomly, that again exponentially increases the number of different types of combinations that you're going to have. So 3 mechanisms: crossing over, independent assortment, and random fertilization are the 3 mechanisms that sexual reproduction and meiosis uses to generate so much diversity in sexually reproducing organisms.